So I would first like to thank the BGRI for the invitation as a speaker in this session. I would also like to thank Kathy Kahn and BGRI for their willingness to make late-minute changes in program. So on Saturday morning, there was the monitoring and surveillance session. And there was a series of presentations that focused on wheat stem rust outbreaks in 2013 that were very unusual. And after that meeting, it became obvious that that information should be shared with this larger group. And so I have only the spokesman and have taken the privilege of taking information from global collaborators to tell this story of what's happened in actually just the last few months. And it's a very important story to tell. So this is just a quick outline of my presentation, and So in terms of background, the last presentation did a good job of kind of giving a background. There was a number of localized important epidemics or outbreaks in this last year. The first one, as alluded to by the last speaker, was in Ethiopia. Also, there was several findings or outbreaks of stem rust in Western Europe, which really hasn't seen stem rust for 50 years. And so this is another remarkable finding. Stem rust was also reported in both Poland and Finland. So I want to talk about these stories. So the first story that I want to focus on is Ethiopia. And so what I'll be talking about is really on the groundwork initially by Dave Hodson and the group there in Ethiopia and the excellent work that they have done. Background, again, what you saw in the last session, the last speaker, was that due to the yellow rust epidemic in 2010, there's been a massive change in the varieties grown in Ethiopia. And a predominant one that is now grown is Digalo, which has good resistance to the UG99. It's on 30% or so of the acreage. And it's been a big contributor to the success of wheat production this year. But it's also a big factor in the stem rust epidemic. So the timeline of this epidemic is outlined here. And briefly, I want to show that we went from a normal season where there was really no stem rust at all through early October. Now, Dave Hodson and his crew were out making regular surveys. And until into October, there was the usual suspects, members of UG99 race group. Nothing new, but for the first time in early October, on Digalo, he saw it become susceptible. And that was the first time it had been seen. By November, middle of November, that had exploded into an epidemic. And in a matter of a few more weeks, the wheat crop in those affected areas were essentially dead. We saw this same picture just a moment ago. And I think for me, it was remarkable, because if I look back in my history books and look at pictures of the epidemics in North America in the 1950s, these dead wheat fields look identical to what happened then and what Norman Borlaug responded to. So the yield losses in these affected areas were up to 90%, average 50% in infected regions. This shows the regions in which that was affected, and you can see the large green dots as the place that had the most highest stem rust incidence. There was a rapid response. It included spore dispersal modeling alerts from colleagues in Cambridge University and CIMMT, extensive sampling. Rapid samples were distributed to many different laboratories for analysis, extensive crop loss assessment, and farmer, regional, local authorities giving control on advice. Now, the second part of this story is to talk about the pathogen side. What have we learned in the last 3 or 4 months about the pathogen that caused this epidemic? And so I'm going to be talking primarily about the data that came from out of the USDA CDL lab in St. Paul, but samples were also sent to the GRC and Mogen's lab in Denmark. So just to give you, I show this slide, it gives you an idea of a timeline. We got in St. Paul samples arrived that span the time from before the epidemic to the peak of the epidemic. So we had the ability to look at what happened. And this is the standard protocol of processing samples, going from field collections to pure isolates so that you can do race typing and genotyping. What was different this time was not only did we use on the left-hand side the standard differential set, but also was used an extended differential series. And I just wanted to show this slide to give you an idea. If you look at the blue boxes, those were usual suspects that we see in isolates. And those were the early time points. The red boxes is when the new race appeared and where it appeared. And this new race has been typed out to TKTT. Now, the next part of this story is to talk about Western Europe. So here in Germany, stem rust was first detected in June. And as I said, it hasn't been seen for 5 decades. So samples were, and it was both on winter and spring wheat. Samples were collected and distributed to collaborators. Mogens also found samples in Denmark. There were surveys done, but no other stem rust was detected in the southern part of Europe. And this shows the areas at which stem rust was found in Germany. And you can see that it extends over a broad area. It isn't a single spot. So when pathotyping was done, these are the 4 main pathotypes or races that were found. And the point I want to make here is that the second one there is our friend TKTT. So we see the same race that was found as predominant epidemic in Ethiopia. We also found it in the outbreak in Germany. So work was done at the GRC by Mogen's group. He race phenotyped the 2 Danish isolates and showed that those were also this new race, TKTT. He also had collections from a number of other places, from Lebanon, Turkey, Iran, Egypt, and also collections from Ethiopia. And he showed that those collections also contained the same race, TKTT. So we wanted to characterize further PGT, this new, quote, new race, PGT, and understand more about it. So continued to look at pathotyping. And the idea was, is what we're seeing in Ethiopia, in Germany, in Denmark, in Egypt, in Iran, are they all the same? And so a preliminary study of looking at 2 isolates from Ethiopia, 2 from Germany, and 1 from Turkey was examined across 163 lines to look at avirulence virulence phenotypes. The Ethiopian and Turkish isolates were phenotypically identical across all of these lines. However, the German isolates were identical to themselves, but they were different from the Ethiopian and Turkish on these 4 genes. So you can see that the line carrying gene 33, Ethiopia and Turkey both was resistant, and there was an intermediate susceptible reaction in Germany. You see the same pattern with 7A, T3, and 45, where both Ethiopia and Turkey are resistant, and the Germany is susceptible. So this shows that on a phenotypic level, that these isolates aren't the same. So there's variability in the phenotypic pathogenicity of these isolates from this small sample set. So the next thing we wanted to do is we wanted to look at genotypically, what do we see with this population? And so to do this, used the custom-made Illumina PGT SNP chip that contained over 1,500 markers and ran principal coordinate analysis on the data. So this is a 3D graph of the principal coordinate analysis. And so coordinate 1, and so in principal coordinate analysis, it looks at variation based on different vectors or coordinates. And so the first coordinate is and so I've plotted here the 3 coordinates. And these 3 coordinates represents 85% of the variation within the population. And I think it's very obvious to you that there are 2 very different types. And they've called them type A and type B. So in the cultures from Ethiopia, we have 2 distinct genotypes. These 2 genotypes are different at approximately 170 loci. Okay. So the next question is how does the Ethiopia compare with the isolates from the other countries? So if we look at isolates from Turkey collected in 2012 and 2013, all of those fall in the type A. If we look at the Germany samples, we find representatives in both type A and type B, but they are variants within these classes. So how susceptible is our current wheat to this new TKTT, this new race? So in Herculean effort in the last month, Eugen and others in the CDL has screened germplasm to get an idea. And I just want to give a quick summary of the data that they have here, that they have. And so they have screened normal germplasm that was put out in regional nurseries in this last year. And this represents over 1,000 different lines. And what I've done here is this is what's resistance to UG99 race group, the specific TTKSK member of that race group. And you can see that this populations, you range from 11 to 40%. In the next column here is what is resistant to both UG99 and this new race. And you can see that a significant proportion of these populations show resistance to both. That's the good news. There is resistance there. So in summary, I wanted to quickly highlight what we just talked about. So we had 2 events. We had a localized epidemic in Ethiopia that caused severe damage in specific regions. We saw stem rust for the first time in Western Europe. And this race is TKTT. What do we now know about this new race? So the first and most important thing to know is it's not a member of the UG99 race group. It is not new. It was first found in Turkey in 2005, but it is fairly broadly distributed. We've seen it in Denmark and through the Middle East and all the way up into Germany. We also know that this race is not a single entity, but there's variation both at phenotypic level and genotypic level. And the take-home message that I want you to walk out is, first of all, in these incidents, there's been a local and global resources that were successfully mobilized to respond to this new events. Live collections were quickly made and distributed globally to be analyzed. Data was quickly shared, and data phenotype was compared and confirmed at multiple labs. And the current wheat germplasm contains resistance to both UG99 and this new race group. And of course, this is a group done by many people, and I would like to acknowledge